Hi everyone, my name is Praveen and I'm currently a junior here at Hawken, and this summer I had the excellent opportunity of researching at the Case Comprehensive Cancer Center with Dr. Jacob Antunes and Dr. Satish Viswanath. And as Anthony said, my project title is Identifying Regional Lymph Node Metastasis on Intensity Characteristics of Perirectal Fat from Magnetic Resonance Imaging in Rectal Cancers using T2 weighted cells. But it's a lot simpler than it sounds. All right, so starting off with some quick background information. So currently every year in the United States, there are 150,000 new colorectal cancer cases each year, and they have a typical mortality rate of around 11.2%, making it the second most deadly cancer in the United States. So currently, some of the problems that patients face is that they undergo 20-year-long treatments with no end success, and oftentimes they have to go through chemoradiation therapy for 10 plus years. Now, something that can actually help these patients is the earlier characterization of metastasis. Now, you'll see in the bottom right of the screen that there's a sort of graphic of furthering stages of cancers. Now, metastasis occurs when cancer spreads from the original place, like the prostate or the rectum, through lymph nodes to other parts of the body. So that was the main thing that we were trying to find in this research, whether we could determine whether a patient had metastasis earlier on in their characterization of cancer. Now, the key imaging tools that we used were MRIs, or magnetic resonance imaging, from radiologists. And moving on to the second slide, the objective goal and the clinical goal of the lab was to characterize metastasis earlier in order to create more individualized and proactive treatment plans for colorectal cancer patients. And the question posed was whether we could use computerized imaging programs and to distinguish metastatic cancer from non-metastatic cancer in order to create better plans for patients. And we hypothesized that using these very computerized features and computerized methods from the perirectal fat region of patients, we could determine whether a patient had metastatic cancer and therefore improve their plans when regarding chemoradiation therapy. And specifically for methods, we used two computerized programs. We used MATLAB, which is a medical imaging and coding program similar to Python and Java, to actually implement the code and test our statistics. And we used another program called 3D Slicer to mark up MRI images and to sort of distinguish different parts of the rectum. And in determining metastatic cancer from these programs, a healthy rectum, or our control group, was defined as one of a prostate cancer patient, as none of these patients actually had metastatic cancer. And going into the second part of our methods, the control set that we were given 75 prostate cancer patients from the university hospitals, none of whom actually had metastatic cancer. And we were also given 50 rectal cancer patients. And going into the experiment, we didn't know the status of these patients. So our goal was to determine whether they had metastatic or non-metastatic cancer. And using, on the bottom, you'll see two graphics. So what we did was, using the 3D Slicer programs, we created annotations for each of these patients. And there are three important parts of the rectum to know. So in the bottom right, you'll see the yellow part, which is the actual rectum of the patient. You'll see the green part, which is the rectal wall of the patient. And you'll see the blue part, which is the perirectal fat. Now, specifically in testing metastasis, when a rectal cancer patient has metastasis, it spreads towards the perirectal fat region. So it spreads from the yellow and green parts through lymph nodes to the blue part. So that is why we specifically tested the perirectal fat region using statistics to determine if a patient had metastatic cancer or not. And to actually determine if a patient had metastatic cancer, we extracted 4 important statistics from this region. So we extracted the mean feature value, the standard deviation value, the kurtosis value, and the skewness value. And using the MATLAB program, as aforementioned, we use these values to compare them from the rectal cancer standpoint to a prostate cancer standpoint. And if the rectal cancer patients had at least a greater than 0.05 statistical value or statistical significance, they were denoted as a metastatic cancer patient. Now, after compiling all of these patients into matrices, as well as their statistics into matrices, we were able to determine that 4 out of our 50 patients were determined to have metastatic cancer. And we also compiled them into box plots comparing prostate cancer patients to non-metastatic rectal cancer patients to metastatic cancer patients to kind of see where everything changed. And the most important statistic we realized was actually the mean value, as the other three statistics were mainly used to prevent the presence of outliers, as well as like skewed data. And these are the box plots. And we saw that the values of the means were between 100 and 700, and the other three statistics were all below 7. And in conclusion, we see that our experiment was actually a success, as we verified with the pathologist after the experiment that these were the patients with metastatic cancer. And it is very important, as we can now characterize their cancer much earlier and set them on a more proactive treatment plan so that they don't have to undergo chemoradiation therapy for 15 to 20 years after the initial characterization of rectal or prostate cancer. And specifically, these plans in the future can be used to not only apply to prostate and rectal cancers, but also future and other types of cancers like breast cancer, for example. And the future goals of the lab are to implement this on a widespread clinical scale all across the northeastern Ohio and greater Cleveland area, so that we can help patients recover from cancer earlier and even lower the mortality rate of colorectal cancers. And I would like to thank my mentors, Dr. Jacob Antunes and Dr. Satish Viswanath, for spending lots of time with me this summer and helping me with my presentation. I would also like to thank Dr. Chick and Mr. Anthony Sloan for specifically helping me tailor my presentation and help me with the overall research project. And I would also like to thank Hawken for giving us the opportunity for researching at institutions like Case Western. So thank you all and have a great rest of your day. And if you have any questions, feel free to stop by my Zoom later today. Thank you.